How this live online virtual tutoring is going to work for you guys. So Professor Grigoriev should have talked about this in lecture with you already. But because this is my first video with you guys, I'm just going to review it again, just so you know exactly how you could best utilize these facilities for yourself. So as I mentioned in the beginning, all these videos are broadcasted live for you guys, meaning you could attend these videos live as well. That would allow you to not only listen to the material, but also provide your feedback and ask specific questions that you want to go over. Now, that is something I'd utilize a lot, because that would allow you to tailor all these videos and all this material to the topics and concepts that you want to review. That would help you guys a lot, and I advise that you use it. And how would you do that? How would you utilize that live session? So in order to sign up, Professor Grigoriev should have shared this Google spreadsheet with you guys already. So every week I add my times and dates that I'm available to provide a tutoring session. And in this column, or rather these columns, you have to fill out name, your email, and the concept and topic that you want to review. This would allow me to better tailor the video to whatever you guys want to review. And not only is the video going to be live, but you could also watch it afterwards, like you're probably watching this video right now. And how would you do that? So Professor Grigoriev should have also shared this Google spreadsheet with you guys. So after every live stream is done and completed, I'm going to be posting its link on the link column right here. Once you click on the link, it should take you to a YouTube video for that specific topic that was covered that day, and you could watch it in your own free time to review the material afterwards too. So now that that is out of the way, let's start going over what I'm actually going to be covering in these videos now. So let's take a look at your syllabus right now. So this is the schedule part of your syllabus. So in the previous week, you guys covered this material, right? And your second exam is coming up. So let's start sort of at the beginning. I'm going to start at animal body and homeostasis. Why? Because that's a huge topic in Bio 102. And that's probably going to be very heavy on the second exam too. Because it's so huge, it's sort of a foundation of all the other material that's coming up afterwards too. So if you understand this properly, it's going to make it easier to understand the material that's upcoming too. So without further ado, let's go straight into that chapter to review it better. And let's start with the slides. So the slides start off with a diagram of, well, what is this? We're looking at, let me pull out my laser pointer or pen, cells, and something called extracellular fluid. So we all should know at this point what a cell is, right? It's a basic unit of life. Well, what is this extracellular fluid term? Well, it has the word cell inside it, right? So it has something to do with cells, but it's also fluid. So we've all probably heard that our bodies, human bodies themselves, are approximately 70-something% water, right? That water is composed in this extracellular fluid portion. Not only are we a mass of solid material, like our cells, but we're also a mass of a lot of fluid. And as the textbook and the lecture slides probably detail further, our cells need this fluid to survive, because these cells themselves are actually living material. And like us, they need to exchange material with their environment. They need to take in nutrients, food, resources, and excrete waste products to survive properly. And that exchange is better facilitated with fluid. Now, there's two types of fluid here. We have our interstitial fluid and our plasma. So interstitial fluid is the fluid that's literally right next to our cells. It's touching our cells. And that plasma is your blood. So what's the difference between interstitial fluid and plasma? Why do we have two different things? Well, later on, you're going to talk about the circulatory system, and you're going to talk about the importance of blood and its flow throughout the body. So interstitial fluid is just fluid that's sitting right outside your cells. It doesn't flow that much, which is okay, but like you want your body to be able to communicate with other parts of its body, right? You want material from, let's say, your fingertip to be able to flow through to your toe. And if the fluid is not flowing from here, any material exported by this cell right here won't flow and reach another part of the body, let's say this cell, right? That's bad. You want your cells to be able to communicate with each other in that sense. And in order to do that, you have these little highways of fluid flow, which are your blood vessels. So, for instance, let's say in this blood vessel, your blood is going to be flowing downwards. And how's the material from the cells going to get into your blood vessels? Well, we know that your interstitial fluid also exchanges material with the blood vessels too. So how does that happen? How does material just flow from cell to interstitial fluid to plasma? Well, the material has to diffuse across these membranes. And diffusion, as from chemistry, you guys should remember, occurs from a high concentration of material to low concentration of material. So let's follow one specific waste product. Let's say a waste product that our cells make is carbon dioxide, right? So over time, this cell right here builds up carbon dioxide inside itself. As time progresses and the carbon dioxide builds up inside the cell, the concentration of carbon dioxide inside the cell increases quite a bit, right? According to diffusion, material flows from an area of high concentration to an area of low concentration. So if we say that the inside of our cell is highly concentrated in carbon dioxide and the outside is not, then that carbon dioxide is going to flow out of the cell into the interstitial fluid. Over time, as material and carbon dioxide flows into the interstitial fluid, the carbon dioxide concentration in the interstitial fluid will increase. And in our plasma, we don't have carbon dioxide present yet, right? Therefore, the plasma has a low concentration of carbon dioxide at that moment. So over time, we know that material diffuses from an area of high concentration to a low concentration. So what do you think is going to happen? Over time, your carbon dioxide is going to flow into your plasma, right? And that flow of material down the concentration gradient allows your body to transport material and communicate with itself, which is imperative for its survival, right? Now, the next slide goes over simple vertebrate organization. So there's many cavities inside our cells, right? We're essentially a hollow tube. And what's the point of this hollow tube? Well, you have different compartments in the body that do different things. And the main compartment is your digestive tract. What does it mean to have a hollow tube through yourself? So it practically is what it sounds like. You essentially have a hollow tube going from your mouth to your anus. That tube is continuous. So you've taken food here, gets processed, doesn't get inside your body, stays inside that little tube right there. And after processing, it just gets excreted right back out. So the material you eat, the solid material, never really gets transported inside your body. It stays out there. Now this slide talks about a couple of different types of cells and how to come up with those cells in labs and stuff. So you've all probably heard of stem cells right now. It's pretty big in research right now. And you know that it's possible to utilize these stem cells to grow any type of cell that you want, any type of tissue that you want, any type of organ that you want. Because of that, they're really useful for us, but it's also useful for you guys to know how those cells work, how those cells grow your tissues that you want. So there's many types, but this diagram looks like it's going over embryonic stem cells. What does it mean to have an embryonic stem cell? Well, as the name has inside it, it's from an embryo. So you're utilizing a fertilized egg to begin with. That fertilized egg, we know, has the potential to differentiate into any type of cell, any type of tissue that we want it to. So what you can do is harvest some of these cells from here, from your fertilized egg cells, and place them in an environment that'll allow them to transform into the tissue that you want. Say you want your cells to turn into muscle tissue. You could place those cells in an environment that'll help them differentiate into muscle tissue. And over time, in your petri dish or wherever you're doing this experiment, you're going to end up with a lot of muscle tissue ready to use, implant, or whatever you have intended for that muscle tissue. Because of that, embryonic stem cells are really useful for us in research. And the slide, and probably the textbook too, which you guys should be reading, it gives us these two vocab terms, totipotent and pluripotent. What do those words mean? So the suffix in the term, potent, means ability, right? The prefix, the toti and pluri, is different between the two. So toti means that that cell has the ability to divide into whatever cell that you want it to. So over time, these embryonic stem cells, starting from the left right here, divide and differentiate into the different tissues that they're becoming. So these cells in the beginning, these fertilized egg cells that have the ability to turn into any type of tissue that you want, are totipotent. Why? Because they have the total potential to become any cell they want. Over time, as they start to differentiate, they lose their differentiation ability. Instead of being totally potential to differentiate into any type of tissue, they can plurally differentiate. They have less choices to become different types of tissue. That's why it turned into a pluripotent stem cell. That's the main difference between those two vocab terms, which are pretty specific to embryonic stem cells. But going into the next slide, so this slide is a cool diagram that puts what we've talked about so far in perspective pretty well. Why? Because we're looking at the hierarchy of the human body, starting with the smallest fundamental unit of life, the cell, right? So we know that our body is composed of cells, but like what does that actually mean? Like, what allows those little tiny microscopic little cells to make up and compose our body and to allow us to do everything that we're doing right now? Well, those cells are actually organized in these functional units. And those functional units are furthermore organized into even larger functional units. And those, guess what? Those larger functional units are also organized into even larger functional units. What does all of that mean? So looking at this diagram here, you have cells, which are the smallest fundamental unit of life. Multiple cells organized to make a tissue. Multiple tissues organized to make an organ. And multiple organs combined together to make an organ system. So the example that this slide gives is of cardiac tissue, which is composed of cardiac muscle cells, right? Multiple of those cells combine to make your cardiac tissue, which we'll talk about in detail why it looks the way it does in a few slides. But that cardiac tissue, many of those tissues combine to make your organ. which cardiac tissue we know makes up the heart, and many of those organs combine to make an organ system, which the heart is composed of the circulatory system. So all our organ systems and organs fall into different hierarchies that yield different organ systems that allow us to be a total organism as a whole. So these slides are basically going over what we just talked about, right? So you have different types of cells that combine to make tissues, that combine to make organs, that combine to make organ systems. Tissues. And here we have a cool schematic of the different types of tissues. So I briefly mentioned that there's many different types of tissue that allow us to make many different types of organ systems, right? So here you have the 4 main types of tissue that you should know for Bio 102. So looking at these images right here, you have connective tissue, epithelial tissue, muscle tissue, and nervous tissue. So connective tissue, as the name implies, is tissue that connects one tissue to the next tissue. We'll see more detail about that in a few slides. Epithelial tissue is very densely packed tissue that is generally used as barriers between other types of tissue. So the main type of barrier that you could think of is like the skin. Muscle tissue is pretty self-explanatory, and so is nervous tissue. Nervous tissue makes up all the nerves and many other different parts of the nervous system. So let's go over these in more detail in the next couple of slides. And that starts with the connective tissue. So this diagram right here shows many different types, the different types of connective tissue that you're responsible for knowing. So here you have your loose connective tissue. As the name implies, it's loose. It's very wobbly, no solid structure. And its job is to connect different organs to each other. So essentially what I'm going to be doing in this slide is practically reading through this. Actually, that's not smart to do. I should just refer you back to this slide because there's a lot of memorization on here. You guys should be familiar with the different types of connective tissue and its function. Why is it the way it is? An interesting fact, though, or an interesting thing about this is that your bone and your blood, the two that I just circled, are actually your connective tissue too. So all the other ones are way different from it, but bone and blood are still connective tissue. Even though bone is completely solid and blood is completely liquid. How's a liquid a tissue? That's interesting, right? Well, it's a tissue because blood is not only just empty liquid, right? It has a lot of cells inside it. We have your red blood cells, the white blood cells, leukocytes, platelets. Platelets are not actual cells; they're parts of cells, but like still parts of cells, right? All of those are living things because there's a lot of them in there, combined together, interacting with each other. Even that fluid solution of cells is considered a tissue. And it has a lot of functions, right, which you could go over in this diagram too. Let me see. So I actually took Bio 102 at NYIT, and at NYIT, we had a different textbook. And from time to time, I'm going to refer back to it because I love the diagrams in my textbook. So I found a cool connective tissue diagram in here. Yeah. So this gives an example of a knee joint of a dog, which, yeah, looks like a dog. And here you have all of the different types of tissues that you saw in your lecture diagram placed into perspective. So you have your loose connective tissue right here that's connecting the skin to the muscles and tissue and bone underneath that skin. You have your fibrous connective tissue that's connecting, which is composed of the tendons and ligaments, which are present in your lecture slides, right? These connect muscle to bone or bone to bone. You have your bone, obviously present right here, right? You have your adipose tissue, which is your fat tissue, which is also connective tissue. Cartilage also present right here between the bones and right next to it. You have your blood and your blood vessels too, right? So all of these different types of connective tissues have to interact with each other in order to allow the organism to function properly, to maintain proper order. What does that sound like? Homeostasis, right? You see where we're getting at in this chapter? All right, back to the different types of tissues. 6 types of connective tissue, lots of text on this slide. Go through it, be familiar with it. You don't have to know everything word for word. I wouldn't. I'd focus more on the diagrams, like understand what these diagrams are about, what they're talking about, be able to recognize what you're looking at. You don't have to know facts off the top of your head like that. Understand the material more. So again, text slides again, right? We talked about blood, why it's important for us. Know that. Don't memorize text word for word. All right, next type of tissue is muscle tissue, right? So this diagram I like quite a bit because it pretty much summarizes all the different types of muscle tissue that you have. So starting on the top, you have your striated muscle right here. And your striated muscle is called that because you see these little striations in it right here, right? You have this one line followed by a light line, followed by another dark line, followed by a light line. Those striations allow that muscle tissue to contract. And you'll see that more once you do muscle in more detail. But essentially, what you should know about this muscle tissue is that they're really long cells. Like each cell is almost the length of the full fiber. So here you see an entire muscle, right? Each cell is probably running from here, this end, all the way to the bottom right there. And you have a lot of those really thin fibers of cells bundled together, excuse me. And each cell has, because it's so large and long, has multiple nuclei in it. This is quite unique, right? We don't ever see that in any other type of tissue. Only muscle cells and striated muscle cells are multinucleated like that. The next type of tissue it talks about is cardiac tissue, which is also sort of striated, right? Some textbooks call it pseudostriated. That allows it to contract the same way that our muscle cells, skeletal muscle cells contract. However, one main difference between skeletal muscle and cardiac muscle is, it should be obvious, that your skeletal muscle is voluntary. So I'll put that right here, voluntary. I'm typing with the mouse. Apologies if its handwriting's bad. Voluntary. And this is involuntary. I won't try to attempt that again. So that's a huge difference between your skeletal and cardiac muscle tissue, which is good for us, right? Because we don't want to voluntarily control our heart. Why? Because our heart's important to function properly. If we voluntarily, accidentally changes the heart rate when we don't want to, then that could be detrimental. That could get us out of homeostasis, cause disorder, which leads to disease, which is bad, right? Now the last type of muscle tissue is smooth muscle tissue, which, as the name implies, is smooth and doesn't have any striations in it. Now, because it doesn't have striations in it, the way it contracts, the way it functions is different from your skeletal muscle tissue. And most of your smooth muscle is also involuntary. Why? Because it mainly makes up all your internal organs, other than the heart, which obviously has its own tissue type. So these are your 3 main muscle types. See how there's very little text on this slide? We talked through these diagrams. This is what you should be doing with this material. Even when you go through your textbook, which you all should be going through, read through the material, yes, but don't just read through it. Try to summarize that material in your own words. Process it. Be an active reader. Be an active learner. Once you process that material, you're ensuring that you're understanding the material that you're trying to cement inside your brain. And that's a great way to make sure that you actually got the material. So I recommend you continue to practice like that. Moving on to the next type of tissue is nervous tissue. Now, your nervous tissue is composed of many different types of cells. It looks like we have a cool diagram of the main type of cell right here, which is your neuron, right? So our neurons are our main type of cells in the nervous system, and they're responsible for communicating information from one part of the body to the other part of the body. Now, we said it's possible to do that through your blood, right? However, this is faster and is the fastest type of communication your body can participate in, which is why you have an entire system dedicated to it. Not only is your nervous tissue composed of neurons, though, but you have a lot of other different helper cells surrounding it too. Those helper cells are glia, and their responsibilities are to make sure your neurons, which are like so specialized, so fast in their function, those glia ensure that your neurons function properly, because if they don't, your body can lose a lot of communication from different parts of itself, which again is a way that it gets dysregulated, causes disease, which is bad for you, right? Do you get the theme about this? All of these different types of tissues are maintaining body order, regulation, something important in biology. So let's look at neurons in more detail. This diagram is pretty cool. Why? Because it's a zoomed in view of a neuron. And there's a lot of text on here, but actually don't pay attention to the text. Let me talk through it first. So your neuron is composed of many different parts. Let's start on the left with this giant yellow blob with projections coming out of it, right? So this is the cell body of the neuron. How do you know that? Because it has its nucleus in here. That circle is terrible, but you see where the nucleus is, right? Not only does it have the nucleus there, but it has all the other different organelles of the cell present in that little section right there too, right? Now, from that little cell blob, you have projections coming off, right? These little hand-looking things. These are called your dendrites in a neuron, and what these dendrites are responsible for is catching a signal from another cell. So we mentioned that neurons are responsible for transmitting information from one part of your body to the next. How do they do that? They catch electrical signals from electrical or chemical signals from one cell and pass it on to the next cell. And they do it really fast. And in order to catch the signal, these dendrites project out into their environment, hoping to reach the end of another cell so that they could catch a signal from that cell and pass it on to the next cell. So once the signal goes into the dendrite, it gets processed in the cell body, which is why you have all your organelles here, and then gets passed down through the axon into the axon terminal, this little end right here, where you have more projections, right? So here you have your input, and at the axon terminal, you have your output, kind of like an electronic device, right? So talking about smartphones, which all of you probably have and are probably addicted to, your smartphone also has an input and an output, right? Say you're using, I don't know, your music app, right? You press the play button and the music starts playing, right? So the play button, is your input, right? Once you press it, you get an output, the sound, the music that starts playing. Same thing here. Once you get an input signal through your cell into your neuron, you're eventually going to get an output out of the back of that neuron. And that's important. These neurons are only one directional. A signal can only go from the dendrites to the axon terminals. That's key for neurons. And in the middle of it, we also see this little long thing, right? So this long thing is called the axon, and it's sort of like a highway for that information to travel down. And on that highway, you see these little blobs that are these white blobs, right? So what are these white blobs? They're essentially blobs of fat that coat the axon, called your myelin sheath. And they coat the axon to provide insulation to the electrical signal that's being passed down, sort of like a wire, right? So all wires are insulated. Why? To prevent loss of the signal from the metal wire that's actually conducting that signal. Same thing here. You want to ensure that that signal is passed without impedance. Not only does it prevent impedance, but it also allows the signal to be transmitted a lot faster. So those two key things, maintaining the integrity of the signal, but also maintaining the speed of the signal, is what your myelin sheath is responsible for. So we just talked about that. Insulates your axon, speeds the signal. Same thing. Numbers are not that important. So we talked about this briefly, right? So we talked about many different types of tissues. Your tissues compose your organs, and your organs eventually combine to make organ systems. Now, the main type of, the first type of system that your slides talks about is the nervous system, which is cool to go into right after talking about nervous tissue, right? Because your nervous tissue combines together to make your entire nervous system. And that nervous system is composed of your brain, your spinal cord, and all of the different nerves coming off of those two organs, right? And we said that your nervous system is responsible for communicating information from one part of the body to the next, right? So that's what this diagram is essentially showing. You get information into your eye. Let's say you see like a terrifying lion ready to pounce at you, right? What would you want to do? You'd want to run away, right? And you need to use the muscles in your leg to do that. Once you see that lion, that's your sensory input. That input is processed by your brain. You think about that lion, about how terrifying its teeth look, about how it could maul you, and you process that information to come up with the idea of running away, right? Once you come up with that idea, you send that information down to your muscles, which are your motor structures. Those muscles get ready to run away from that lion. That is essentially how the nervous system works. Takes an input, processes the information really fast, and then produces an output. Doesn't have to be a motor output. It could be anything. I'm talking, you're listening. All different types of outputs. Homeostasis, balancing act. Yay, elephant can bounce on a beach ball. So what's the point of this picture? So it's showing an elephant balancing, right? That's essentially a summary of what homeostasis is. There's so many factors, so many different things going on in our body. We looked at the smallest fundamental unit of life, a cell, right? A cell is tiny. And because it's tiny, we humans are composed of thousands, probably millions of those cells. At those millions of those cells are all communicating with each other, communicating with their environment. And all of that communication has to be done in a proper balanced way. That is homeostasis. And homeostasis, as the definition gives you, is the dynamic constancy of the internal environment of an organism. What does it mean to be dynamic? It means, it's sort of a contradiction, right? Dynamic means changing, constancy. Like, what does that even mean? How does it even make any sense? Well, think of like a, actually, we'll get back to that later. So how is this homeostasis regulated? So the main different type, So there's different types of ways that organisms maintain homeostasis. There's different types of organisms, and they have different ways to deal with changes in the environment. So the first type are regulators, and the second type are conformers, as the slide says. And what do these different types of organisms do? So a regulator is someone that will maintain internal environment regardless of external environment. So the main thing this talks about is, main example it gives is of body temperature. Say you have an organism that likes to keep its internal body temperature high, close to 100 degrees Celsius. If it likes to keep it in that range, it will always keep it, and it will use its energy to keep it in that range, regardless of what temperature it is outside. That is a regulator. A conformer would be an organism that allows its internal body temperature to change based on whatever environment it's in. So say you're in a hot room, that's 50 degrees Celsius, your internal body temperature will get to 50 degrees Celsius over time, right? Because you're sitting in that hot room. But if you step out into, let's say, a blizzard and it's like 0 degrees Celsius outside, that organism will also allow its temperature to reach 0 degrees Celsius in order to conform to whatever environment it is in. That's the difference between a regulator and a conformer. Why is this important to discuss? Why is this difference important to discuss? Think about energy. So your regulators are using internal energy in order to maintain whatever consistent environment they're maintaining. Whereas conformers don't have to do a lot, right? Because it's the environment that's changing them. They just conform to whatever conditions there are present in the environment. So it takes a lot more energy to be a regulator than it does to be a conformer, which is important for us to know. What type of organism do you think these guys are, these little penguins then? They're actually regulators. And why do you know that? Because they're trying to maintain their body temperature by huddling together like this. And it's crucial for them to do that because a regulator cannot be a conformer and a conformer cannot be a regulator. They can't switch like that. So if you're a regulator and the environment's becoming too harsh for you, you really have to do something to regulate your internal environment. And this is sort of like a last resort that the penguins have to go to. If they're getting too cold, they have to huddle together to produce enough heat to maintain their body temperature because they can't conform to the cold external environment. They can't let their body temperature drop because that would be dysregulation and that could lead to disease, which is bad for them, right? So how does homeostasis work? Well, feedback is one key way a constant environment is maintained. And this will help make more sense of the contradiction that we saw, a dynamic constancy. So here we have a little network of a stimulus yielding a response. And that response doing something to help change the processing going on, right? So let's start with the stimulus because we always start there, right? That's your input. Once you receive an input, you process it with your sensor and your integrating sensor. That integrating sensor feeds that information to the effector, which makes a response. And in some cases, that response can go back and turn off the processing because if you have enough response, you don't need to make more of that response, right? That type of feedback is called negative feedback because your response is turning off the production of more of that response, which is useful for us, right? Because it prevents the waste of energy. If you have enough response, why do you need to make more? You don't want to make more, right? So you could stop utilizing the energy that you're utilizing in order to make that response. An example that I like to give for this is a thermostat. So let me pull up a new slide. Let's draw something. So how does a thermostat work? A thermostat's a good example of a negative feedback loop. So you have your thermostat, right? Which I'll draw as a rectangle. and thermometer. Your thermometer, right? Can I erase eraser? Let's make a better thermometer. So essentially, you have your thermometer, right? Let's assume that's a thermometer, right? Now that thermometer in your house is connected to your heater, right? Heat. Okay, so that heat feeds its information back to that thermometer, right? So review, how does this work? So you have your thermometer, right? It senses that the temperature is getting too cold. It's getting temperatures drop too much. When the temperatures drop too much, you have to kick on that heat, right? To bring the temperature back up. Once that heat is turned on, over time, the temperature will rise again, right? That heat is feeding its information back to the thermometer. And once that temperature rises to a certain level, the heat will stop, right? It will cut off the heat. Why? Because you don't need to heat the house up more. You're just wasting energy then, right? That is a prime example of negative feedback. We use this every day. So do our bodies to maintain homeostasis. So you see how you're maintaining a constant temperature through your thermostat by the use of negative feedback. However, stuff isn't actually staying constant, right? Your heat's constantly turning on and off. This temperature is going to fluctuate a tiny bit in that range, right? So there is change going on. It's just that overall, when you look at the overall thing, there's not a lot of change going on. Overall, it doesn't look like it's changing. That's why it's called a dynamic constancy. Dynamic means at the micro level, stuff is changing, but overall, the organism maintains a constant environment inside itself. So this slide essentially reviews what we just talked about. Examples, internal thermostat, kind of like what I talked about with my thermostat example, right? Now, not only do you have negative feedback where your response actually turns off the processing, but you also have the opposite as well, called positive feedback. And as the name implies, positive feedback turns on that response, creates more of that response. And in some cases, it is quite useful. You need to do that in some cases. Go over this diagram, essentially pretty self-explanatory. So the next thing that your slides go into are how how different organisms utilize energy in different ways. So this diagram is showing you a graph, and the first thing I like to look at on a graph, on any graph, are the axes. So what does the x-axis show? It shows your mass, right? And your y-axis shows mass-specific metabolic rate, which essentially means metabolic rate, right? And what do you see? What's the relationship between the two axes? You see that as your mass gets smaller, right, going to the left, your metabolic rate increases quite a bit, right? What does that mean? Why do small organisms use so much, metabolize so much, and utilize so much energy? Why do they have to do that? Well, your slides have the answer. You should have gone through this already. But pretty self-explanatory, right? Think of that mouse right there. Compare it to the elephant. That mouse is a lot smaller, right? However, both of these two organisms are regulators. They have to maintain their internal body temperature. Let's throw both of them into a freezer. Which one's going to drop its temperature faster, and why? The mouse will drop its temperature faster, right? Because it has less mass, and less mass means that it's going to be easily affected by its environment. More easily so than the larger mass organism, the elephant, right? Because it's more easily affected by the environment, that mouse right here has to do a lot more work in order to maintain a constant environment inside. And that work is where it's metabolizing, utilizing energy. That's why smaller organisms metabolize more. And that pretty much is the end of your PowerPoint. So let's stop here for today. And next video, we'll probably do something similar, go through the PowerPoint again, unless you guys have specific material that you want to cover. And please do share that with me, because that will make these videos a lot more helpful for you guys. Right now, I'm just going through the material, which hopefully helps too. But if you share specific material that you want to go over with me, then I could focus more on that than other stuff. All right. Thanks for watching. Bye.